Hey, welcome back. Today we're going to go through the neuroscience of the cerebellum. The cerebellum is basically what the brain's coordination control system is. It receives input from all the sensory modalities, especially from the spinal cord and the special senses such as vision and the vestibular structures in the ear. And they all project to the areas involved in control of the motor areas, and it integrates the sensory and the motor information on the ipsilateral side. So, in doing so, it regulates all the aspects of movements and the vestibular ocular reflexes, along with motor learning. So, the cerebellum lies right behind the brainstem and the fourth ventricle in the base of the brain, and it's folded in itself with a lot of like folds, and they're called folia. These folia are separated by fissures, and the superior cerebellar peduncle is going to be joined to the midbrain, the middle to the pons, and the inferior to the medulla. There's a bunch of fissures that aren't honestly that important to know, but the important ones are the primary and the horizontal fissure, along with the posterolateral fissure. The spinal cerebellum is going to be made of two parts, the vermis and the paravermis, and those will receive the proprioceptive and sensory inputs. These are responsible for smoothing and coordinating movement by comparing the plan with the movement. So, for example, if you're reaching out to grab something, your brain has already kind of calculated how far you need to reach and how much like tone in the muscles the hand needs to be in order to grip the cup or to hold out the arm all at the same time. And people who have a problem with the cerebellar, I'm jumping ahead of myself, but like people with a problem with the cerebellar, or people, especially children who haven't developed the necessary skills and the learning process yet, they're still learning, right? They are jerky and unsure because they don't know how far to reach and how strong their grip strength should be. That's why kids do tend to drop glasses and bowls quite a lot because they're not sure how strongly they need to grip or how much tone in the arms they have to maintain in order to hold, let's say, like a gallon of milk. I mean, don't give children a gallon of milk to hold, but, like a water bottle. So, the vermis is going to project to the brainstem nuclei, and it controls the axial and the proximal limb muscle, so this controls like central posture. The paravermis is going to project to the brainstem nuclei that controls the distal limbs. So this is the one that really controls skilled voluntary movements. So if you played any kind of instrument, that's a skilled, a learned skill, writing, what else? Walking. Those are skilled movements that the paravermis is responsible for. The vestibular cerebellum is in charge of equilibrium and learning-induced changes in the vestibular ocular reflex. So equilibrium comes naturally, like standing, like moving your head. These you don't have to learn actively. But the learning-induced changes are kind of like when you start running, right? There's a difference in equilibrium. When you go on roller coasters, or when you go and fly, there's a change. So these are learned. Like when you go into the airplane, you kind of know like that feeling of takeoff. You're feeling the G's. So these are your learned changes. The cerebrocerebellum is going to be connected mostly to the pons, and this is responsible for planning and programming of the movements. So This is the one that I was talking about, right? So your plan to reach forward for a cup. The climbing fibers are going to be your afferent fibers, and these go through the inferior olivary nucleus. The mossy fibers are the cerebellar afferent from the brainstem and the spinal nuclei. The Purkinje cells are the efferent fibers, and those go to the deep cerebellar nuclei. So here are your climbing fibers. Here's your mossy fibers. Here's your pontine mossy fibers, and the dentate intersposed will go to the thalamus and the red nucleus. The cerebellar cortex is made out of layers, which are the outermost molecular layer, then the Purkinje cell layer. So remember, Purkinje are the efferent, sends the signals away. The white matter, and then the granular layer. The molecular layer is the most superficial, and it's going to be made out of interneurons, which are the inner basket cells, the outer stellate cells, and then the input is going to come from the granular cells. Output are going to be the dendrites and the cell body of the Purkinje cells. So, because that's the only afferent. Let's go back. So this is kind of important. The parallel fibers and the input fibers they're all glutamatergic. So, these use glutamate as the neurotransmitter. The Purkinje cells are going to be GABA neurotransmitters. Okay, so Purkinje layer, they're a large Golgi type 1 cell, and the input comes from the molecular layers along with the climbing fibers and the parallel fibers. And the output are their dendrites that go into the molecular layer, or the axon that go the other way into the deep nuclei. And the basket cells surrounding them are going to be the inhibitory interneurons. The granular cell layer are made out of interneurons that are granule cells and Golgi cells. The Golgi cells are inhibitory, and the granule cells are projection fibers. The fastidial nuclei is near the midbrain in the vermis, just dead center. The intersposed nuclei is going to be made out of the globus and the emboliform, and that's where the paravermal portion of the spinocerebellar projects. So right here, the dentate nucleus is where the cerebellum gets the input from the neocerebellum, and it contains the white fibers made out of efferent fibers that leave into the superior cerebellar peduncle. Okay, so white matter are the arbovitae, and they are afferent fibers which go to the cerebellar cortex, and those enter through the medulla, or inferiorly through the medulla, and medially or middle it goes through the pons and the cerebellar peduncles. The efferent fibers are the same. It's still going to be the Purkinje fibers, and those nuclei are going to be multipolar, and those project through a whole host of systems. The inferior cerebellar peduncle, that's where the ParaAllo cerebellar fibers project to. So here are all the afferents, here are the efferents. There's a lot of fibers. What's more important is that they are concerned with integrating the proprioceptive sensory input with the motor vestibular functions. So what does that mean? It's going to integrate proprioceptive information, and with that, the body is going to use the motor system to compensate for whatever, right? So it's maintaining balance and posture. The middle cerebellar is only a receptive thing. It is going to integrate the information. There's no afferents. Or there's no efferents, sorry. The superior cerebellar peduncle is going to go straight into the midbrain, and here are the afferents, here are the efferents, but it's going to be coordinating the ipsilateral arm and leg. The blood supply of the pons, medulla, and midbrain really comes from the basilar artery and all of their lovely projections. So it's going to be the superior cerebellar arteries, the anterior inferior cerebellar artery, and the posterior inferior cerebellar artery. And all of that leads back from the basilar artery. Okay, so in a disorder concerning the cerebellum, there are no sensory defects because it's not a sensory organ per se, right? It's not in charge of receiving that. It just integrates the information. However, there is going to be some defect in the voluntary control movement because there's slight problems, or slight, depending on how bad it is, right? There's going to be problems with proprioception, so the body isn't going to be able to compensate correctly. However, the reflexes are normal. There's hypotonia due to whatever, like paralysis involved, weakness involved. There's no paralysis, like I said, but hypotonia because there's no information being sent back. So there's no long-term adjustment in the motor response, and there's going to be a defect in the vestibular ocular reflex, which is going to be nystagmus. So if you do the finger test in front of patient's eyes, when you move your finger and they follow the course of your finger, there's going to be like a tick in their eye. Their eyes are gonna like flash to the median, like again and again and again. They can't control it, so it's almost like a tick. One of the signs of the disorder or anything involving a cerebellum it's not a diagnostic criterion per se, but it does give you a lot of helpful hints that this is going to be a cerebellum infarct or whatever, is the Charcot's neurologic triad, which is nystagmus, an intentional tremor, and scanning speech. The nystagmus because of the vestibular ocular reflex problem, intentional tremor because of the problem in coordination, the problem with inhibiting excess movements. There's a problem with tone, and scanning speech because there's involvement with like the Broca's area, Wernicke's area. Okay, so Friedreich's ataxia is characterized by gait disturbance, speech problems, along with heart disease and diabetes. It can be genetic. It can be due to a trinucleotide repeat on chromosome 9, or there could be like a disorder of mitochondria metabolism. It's caused by the degeneration of the nerve tissue in the spinal cord, especially the sensory neurons. So there's going to be a problem with like proprioception. There's going to be a problem with like compensation in terms of like, movement. There is no reflexes present, and there is going to be a certain gait. It's the ataxic gait. Okay, so the cerebellar ataxia is going to be a defect in coordination due to errors in rate, range, force, and direction of the muscle contraction. If the cerebellar cortex is involved, the ataxia is temporary. If the lesion involves the deep cerebellar nuclei, however, the ataxia is permanent. So there's a particular gait. It's called a Lambden gait, and there's going to be dysmetria along with the rebound phenomenon, which is like in most people, if you ask them to do an alternating movement and you tell them stop, they can stop. But in these people, they have to finish the movement before stopping. They can't just stop halfway, right? In adiadochokinesia, that's when you can't do the rapid movement like quickly, or the alternate movement quickly. Decomposition of movement is problems with complex movements. Okay, so how do we test this? We check for the nystagmus, we check for slurred speech, and we test the gait. Sometimes the gait is that bad that, like, you can't get the patient out of the bed, or maybe they're there for a stroke. So you do a finger to nose test along with a heel down the shin. If there is wiggling, like the patient can't touch their finger to your finger, like their finger to nose to your finger, and there's like wobbling around, that's called dysmetria. And the heel down to the shin, that tests like the posture test, really, but clearly lying down in bed. The posture test for a normal, healthy person is standing with your feet closed but your eyes open. If you wobble, there's going to be problems with the vestibular apparatus, right? Romberg's test is going to test the proprioception, vestibular function, and vision altogether. The purpose of this test is to test for whether the lesion is a cerebellar problem or a problem in the dorsal column. So patient stands there with their feet like shoulder width apart, and you ask them to close their eyes, and you gently push at them. So usually when the eyes are open, or not usually, every time when the eyes are open, all the systems provide the input to the cerebellum with some compensation from the eyes. So in a dorsal column tract lesion, so a problem in the spinal cord, the patient is going to lose their balance when their eyes are closed. So they close their eyes and you give them a little push. In a normal person, they stumble a little bit, but they can compensate by like leaning forward, shuffling their feet, whatever, but they stay upright. These people just kind of fall down. There is a problem because this is a problem with proprioception only. There is compensation when their eyes are open. So that means that everything is being coordinated correctly in the cerebellum. Things are being able to be relayed and interpreted. There's a cerebellar lesion when the test is positive and the eyes are open. Eyes are open, you give them a little push and instantly they fall to the ground. There's no compensation involved at all. Okay, so that's it for now. Thank you for your attention, and I'll leave you with a quote from Jean-Marie Charcot. So, symptoms then are in reality nothing but a cry from suffering organs. And as physicians, we better hear that cry.